the things that are in the cortical labyrinth, well, aside from corpuscles, the tubules that are in the labyrinth, of course, are the proximal and distal convoluted tubules and the arch collecting ducts. The medullary rays don't have corpuscles, and they are just straight tubules. So cortical collecting ducts and proximal and distal straight tubules. So there were a couple of lines on either side of a medullary ray, and it was just ID that medullary ray. Okay. So next we were asking about the JG apparatus. And this was just a question designed to test your knowledge of the JG apparatus and can you identify everything associated with it. So what makes up the JG apparatus? What's in the JG apparatus?
Macula densis.
Yep, macula densa cells. What else?
The JG cells and then the extraglomerular
Mesangial cells. Perfect. And what else? The JG, macula densa, extraglomerular mesangial cells. Okay, so those things make up the JG apparatus. So then can you identify those things? Well, here is the extraglomerular mesangial cells right here. JG cells, they're modified smooth muscle cells. So you know that they're in the wall of the afferent arteriole, but you can't see them without an electron microscope. But to identify them, quote unquote, identify them, the afferent arteriole would be indicated. And again, they're modified smooth muscle cells in the wall of that arteriole. And then the macula densa cells, well, what are they? Well, they're modified cells in what tubule? What specific tubule?
distal convoluted.
Yes, the distal tubule. It's a little tricky. It can be straight or convoluted at the point where it comes back to the renal corpuscle. So we tend to just say distal tubule just to be safe. But here is a macula densa right here, that specialized region of cells in the wall of that distal tubule, and you'll identify it because the nuclei are all really close to each other and they're pressed up against the extraglomerular mesangial cells right here. And you can see this afferent arteriole is about to run through the core of all that. So those JG cells in that wall of that afferent arteriole, they'll be all, in close opposition to these other two components. So we were just wanting you to be able to identify one of these components, say like put a box around the macula densa cells, or outline the extraglomerular mesangial cells, or indicate the afferent arteriole, which of course would have those JG cells. Okay, let's see what else. We ask you about we ask you about mesangial cells. And then we ask you about the interlobular artery. We've already covered the mesangial cells, so I'll move on to the interlobular artery. So this is a question testing your knowledge of blood flow in the kidney. And, we've had blood flow questions before, and in the kidney, like the spleen, these are two organs that blood flow is critical to the function of that organ. So it's important to understand the blood flow as it comes into the kidney and goes out of the kidney. Remember, the kidney is filtering 20% of the cardiac output. So that's a lot of blood flow. So in particular, we were seeing if you could ID an interlobular artery. Now, if you can ID an interlobular artery, that's also testing your knowledge of, do you know what a lobule is in a kidney? So a lobule is half of a cortical labyrinth with a central axis of a medullary ray, and then a half of a cortical labyrinth on the other side of that medullary ray. So these interlobular vessels, here's an interlobular artery, and here's an interlobular vein, they mark the boundaries of that lobule. So this is one kidney lobule right here. So one of these interlobular arteries right here were being indicated, and you were asked to ID that vessel. Those interlobular arteries come off your afferent arterioles, which run here in your excuse me, your arcuate arteries. Whew, the interlobular arteries come off your arcuate arteries, which arch, arcuate, hence the name arcuate. They arch right here at the cortical medullary junction. Okay, so this is indication of an arcuate artery right here. They tend to have a lot of connective tissue associated with them here at that cortical medullary junction. So arcuate arteries, even if you don't see much of them, when you see these dense areas of dense irregular connective tissue, that lets you know you're kind of near one of those arcuate arteries, even if that plane of section doesn't let you actually see it. This looks like an arcuate vein right here, and they arch this way at that cortical medullary junction, but what they give off is those interlobular arteries. And if you understand what a lobule is, then you'll understand that that's an interlobular artery and not an interlobar artery, which would be out here in between the lobes, because remember, in the embryo, all of us have lobes, and then varying amounts of fusion occur amongst the species. So in the carnivores, all their lobes fuse, but they still have those interlobar arteries. All right, so that was the interlobular artery. There was a cortical labyrinth question. Let me look at it. So it's just what was not considered not to be part of the cortical labyrinth. So it's just testing your understanding of the components that could be found in a cortical labyrinth. There was a question about peritubular capillaries. So it was which of the vessels give rise to a peritubular capillary network, and that would be up in the cortex where we asked. And again, that was just a question testing your understanding of the blood flow. So these interlobular arteries will give rise to afferent arterioles. So here we have a little hint of an interlobular artery right here. And you can see it's giving off this branch, which is headed right over to this renal corpuscle. So this is the afferent arteriole. And that's the only time I feel, real confident that I'm looking at an afferent arteriole is when I see it coming off that interlobular artery. That afferent arteriole, of course, will feed into that glomerulus, the capillary tuft inside the corpuscle. And then coming out of that capillary tuft will be the efferent arteriole, which is a highly unusual situation to have both an arteriole both feeding into and draining a capillary bed, but the body does that because these arterioles have smooth muscle around them, and then we can control the pressure in that capillary bed both by the afferent arteriole and the efferent arteriole. It's like having a valve on both ends of the blood supply going into that glomerulus. Okay, any questions about those blood supply questions? Okay, did I finish that though? The efferent arteriole gives rise to those peritubular capillaries. So again, your arcuates give rise to your interlobulars, which give rise to your afferents, which give rise to the capillary tuft in the glomerulus inside the renal corpuscle. Then they are drained by that efferent arteriole, and the efferent arteriole gives rise to all of these peritubular capillaries. And if you zoom in, they're all just surrounding the tubules here in the cortex, hence the name peritubular capillaries. Okay, and not all of them have blood in it, so it's hard to ID them if they don't have red blood cells in it, but many of them do have red blood cells in it to help you ID them. Okay, we had a question about podocytes, and this is to test your understanding of how the corpuscle is put together. Okay, so what is another name for the podocyte cell layer? Anybody want to throw that out there?
Isn't it something like the visceral layer?
Epithelium. That is right. That is correct. So the glomerulus has a capsule here. The renal corpuscle has a capsule that surrounds the glomerular tuft. And remember, the the glomerulus kind of invaginated or pushed its way into the blind pouch, the blind ending of that tubular system, the nephron that was forming in the embryo. So when this capillary tuft pushes into that blind pouch, think of it like an elongated balloon, and you push your hand into the end of an elongated balloon. You know, those kind that, you know, clowns twist into cool shapes like giraffes and stuff at, you know, kids' parties. You take this elongated balloon, and on that blind ending, you just push your fist in there, and you let your fist be the glomerulus. So when you push your fist in, then you have that part of the balloon that you're contacting is tight around your fist, but then there's an air-filled space, and then there's the rest of the balloon on the outside. That's exactly what we have here. So this capillary tuft, the layer that's just tightly adherent to the outside of the capillary tuft, that is your visceral epithelial layer, and that is your podocyte layer. But then this is your parietal epithelial layer, and this parietal epithelial layer is continuous with the proximal convoluted tubule when it starts at that urinary pole. And of course, this is your urinary space. So, you know, it's kind of like, you know, when you have the pericardium around the heart, you know, the visceral part is what contacts or lays on the heart, and then the parietal part is what's, you know, separated from your pericardial space. So it's a common theme that repeats itself throughout the body. So we were just seeing if you knew that other name for the podocyte cell layer. Any questions about that? Okay. Let's see. There was a question about the glomerular filtration barrier, and it was just, could you, you know, it's another question testing your knowledge of the glomerular filtration barrier. Do you know all the different parts? And could you identify which of the items listed did not belong to the glomerular filtration barrier? Okay, does anybody want to throw out there what they think is in the glomerular filtration barrier? There was a really nice little schematic in I believe it was in your lecture notes that that showed you the glomerular filtration barrier. And did you go over it in renal phys, the glomerular filtration barrier?
I would think they would have gone over the three components.
Mm-hmm. So if you haven't, go over that schematic again. I could bring it up, the schematic. It would take just a little bit of digging.
While Dr. Jared's thinking, we always said whenever we want to cause a filtration to occur, like in the lungs, where we have two epithelial cells sharing a basement membrane, we also have that kind of scenario in the choroid plexus where we make our cerebral spinal fluid. And then once again, when we go into the kidney, we need to make a leaky vessel. So we take two epithelial cells and they share their basement membrane. So the two epithelial cells themselves make up part of the barrier, as well as the basement membrane that they share. So those are the three components. And in diseases, if you get thickening of that basement membrane or a change, as Dr. Jared mentioned, a charge in that basement membrane, it'll affect the actual ultrafiltrate.
Okay, so here are the lab notes. Can you see them?
Yep.
Actually, the lecture notes. So this is what I was talking about, how the you know, the glomerular capillary tuft just invaginates into that blind pouch ending that is, you know, in that developing nephron in the embryo. So then this is the part that, you know, is tightly adherent to the outside of that capillary tuft. So this is your visceral layer of the renal corpuscle, which is podocytes, see right here. And then this is your parietal layer of the renal corpuscle out here, okay? So that's what we were just looking at here. So this would be your parietal layer out there, and this would be your visceral layer here, and that's podocytes that make that visceral layer of the corpuscle. So that question was just testing your knowledge. Had you know, taken this information here, and could you take a schematic and then apply it to what you see on the slide? Now, here is the schematic that's really helpful to understand the glomerular filtration barrier. So here's just a capillary loop pulled out of that glomerulus, that capillary tuft. Here are the podocytes wrapped around it. Here's the podocyte cell body. Here are their little foot processes. There's little primary and secondary foot processes. You get the little filtration slits in between these foot processes. There's a shared basal lamina that is between the podocytes and the capillary tuft of the capillary of the glomerulus. So, and the capillary itself has a fenestrated endothelium. So you've got your fenestrated endothelium, which is a little bit leaky. And then you've got this specialized basement membrane, which is shared between the capillary and the podocyte layer. And that is what, of course, the filtrate of the blood must pass through. And I'm sure you learned all about charge and protein size and what can get through and what can't in renal phys. And so this urinary filtrate then enters into that urinary space, which is right here. Okay. So we were just asking you which of the following was not part of that glomerular filtration barrier. Okay. Let's see what else. Now we changed, so we were through with the urinary system and we went into the special senses. So there was a question that said select all of the listed components that makes up the membranous labyrinth. So, you know, whether it's a membranous labyrinth question or whether it's a bony labyrinth question or osseous labyrinth question, it's a way to test, have you put the big picture together? Do you understand the semicircular canals versus ducts? So one is bony, the canals are bony, the ducts are membranous. In the, you know, the vestibule holds what's inside the vestibule? The utricle and the saccule. So the vestibule is bony, but the utricle and saccule are membranous. And then the cochlea. So the cochlea is bony, but the cochlear duct is membranous. Okay, so it's just to see if you put that whole picture together and can you select out of a list which components are bony or osseous versus which are membranous or form that interconnected ductwork that connects between both the semicircular canals, the vestibule, and the cochlea. So are there any questions about that? Okay. Let's see. There was a question about the eye, the basement membranes of the eye. So there are a lot of specialized basement membranes in the eye. Can anybody think of where we have basement membranes in the eye? Is that too far out? You haven't been thinking of the eye lately? Okay. So what about in the cornea? Can you remember basement membranes in the cornea? I can change slides. Let me do that. Let me change lessons. Okay, let's see. Can you see that slide there? Okay.
Yep.
So in the cornea, where do we have basement membranes? Well, this is an epithelium. Would you expect that to have a basement membrane under it? You betcha. There's always a basement membrane between an epithelium and its underlying connective tissue. I can think of one exception, but we won't go there. You'll learn about it in neuro. And then back here, so you've got an anterior epithelium and you've got a posterior epithelium, aka the endothelium, which is kind of a misnomer, but the ophthalmologists aren't going to give it up, so we're stuck with it. So here we have this greatly thickened specialized basement membrane here between the corneal stroma and that posterior epithelium. And then we have just a traditional, thinner basement membrane that you can just barely see. Really, we would have to do special stains to get to see it. That's also a basement membrane. Okay. So those are two components of the cornea, our basement membranes. What about this right here? This is the lens. What do you call this right here? It's on the anterior surface of the lens. This is lens epithelium. So this is the epithelium gives rise to these lens fibers. And so an epithelium tends to have a basement membrane. In the eye, sometimes these basement membranes are weird. Like they face the vitreous. Okay, so that's what we've got here. This capsule of the lens is actually a basement membrane. Okay. And then let's see, the choroidal epithelium. Let's see. Oh, the basal complex of the choroid. Yeah, that was one that was listed. So the choriocapillary layer. So if you know your layers of the choroid, you got your suprachoroid layer, you've got your vascular layer. Remember that suprachoroid layer is like, why did we even bother naming it? It's here where the choroid likes to pull away from the sclera. Suprachoroid, this is a vascular layer, which think of it as kind of like the stroma of the choroid. Then you've got your choriocapillary layer right here. And then this is the pigmented epithelium, retinal pigmented epithelium. And between that choriocapillary layer and that retinal pigmented epithelium is called the basal complex. And that's a specialized basement membrane that is shared between the capillary bed and the retinal pigmented epithelium. Okay. And so the one thing that was listed that is not a basement membrane is, and this was to see if you understand the layers of the iris. This is the anterior limiting layer of the iris. It is not a basement membrane. It's just a weird layer made up of these specialized stromal cells, these fibroblast cells, and not a basement membrane. It's just called an anterior limiting layer, but it's not called the anterior basal complex or basement membrane or anything like that. So that was the one thing that is not a basement membrane. Okay. And let's see, there was a question about, could you ID rods versus cones? And that was to see if you understand how we name rods and cones, and have you looked at them, and can you ID them? There were pictures of them in your lab notes where we actually used oil immersion to show them to you. So this is a good one, the herbivore section. So all these little linear lines right here in your outer segment of your photoreceptor layer, these are rods. And let that name tell you, see how they're all linear, they're rod shaped. And then see how these are kind of bulbous or cone shaped. So these are cones. Okay, so cones, rods, cones, rods, cones, rods. They're all mixed in together. So that was to see if you could ID one of those. And let's see. So accommodation, there was a question about accommodation. And that's testing your knowledge of the components of the ciliary body and how they work, how those components work to make the process of accommodation occur. Okay, so what was default? We have our ciliary muscle right here in this herbivore. So these are meridionally positioned. That means kind of along the kind of the long axis of the on a sagittal plane of an eye section. So these are smooth muscle cells, and in default is relaxed. Okay, and that makes sense. Muscles tend to be relaxed by default. So if they're relaxed, then they're they're pulling, they're kind of pulling down, keeping the ciliary body flat, which will keep tension between the zonular fibers, which originate here along what is actually the basement membrane here on the outer, non-pigmented layer of the ciliary epithelium. Right here is a basement membrane. So those zonular fibers are attached to that basement membrane right there, and they span over to the lens, okay? So if there's tension on these guys, these zonular fibers going over to that lens capsule, so from basement membrane to basement membrane, lens capsule to the basement membrane of the ciliary epithelium, then if that ciliary smooth muscle is flat, we're keeping that tension. But if this ciliary smooth muscle contracts, shortens, it will mound up this region right here. It'll cause this connective tissue right here of the ciliary body to kind of become mounded a little bit, to kind of, you know, protrude in towards the posterior chamber. And that's going to happen all the way around the eye, 360 degrees all the way around the eye. And that's going to relax tension on these zonular fibers. And therefore, we're not pulling as much on that lens capsule, and the lens is going to want to round up. Okay. So the question was, what happens for near vision? And near vision is that ciliary muscle contracts, that ciliary body mounds up. We release tension on the two basement membranes, and that lets that lens round up so that you can magnify, you can look closely at things. Default is flat, not magnifying, so you can see far away.
And there's also a, in my PowerPoint on special senses, there's a diagram showing you photo accommodation and the changes in those fibers and in the lens, if you're still a little cloudy on understanding how that works.
And then to go along with that, there was a question about the zonular fibers, what they're attached to. And you had an image to go with it to show you, so that you could decide what those zonular fibers are attached to, okay? So here, I'll zoom in. Let me turn this a little bit. And here are the zonular fibers here. Here is our outer pigmented layer of the ciliary epithelium. Here's our inner non-pigmented layer. And you can see the zonular fibers are coming in and they're attaching to this inner non-pigmented layer of the ciliary epithelium. So that kind of went along with that accommodation question, and it had an image so that you could reference it to see if you understood where those zonular fibers are spanning between. And that was it for quiz 4. Any questions about that? Okay. Let's see if I can figure out how to stop sharing. There we go. All right. That's all I've got. If you don't have any questions, Dr. K, I know you've been distracted trying to help Li Ming, but we went over, you know, what organ systems are going to be included, about how many points for each organ system. I told them you would be sending them information about when to download the exam and when to start it. Of course, we won't be starting it until 8 a.m. on Monday, the Monday after Thanksgiving. And gosh, I think that's about it, isn't it?
I think so. Now, I guess I noticed that we have, they have Wednesday, Thursday, and Friday off. I will try to get to Jacob so that we could have plenty of time to download it. And then I'm assuming that we're going to do the exam the same way we did the midterm. And that is that there'll be some students that will be in person, and there will be
No.
some students that will be at remote sites. I noticed that,
Dr. K, we
Yep.
don't come back at all after Thanksgiving. We'll all be remote.
oh, that's because I was looking here and it says on your schedule, it says, excuse me, it says here you have lecture at 10 for physiology, but that's going to be all virtual then?
Yeah, everything is pre-recorded for phys for the rest of the year.
Oh,
Other
okay.
classes
So
might
then-
be on Zoom.
Well, that's great, because otherwise I'd be showing up Monday morning looking for everybody. So what we'll do is we'll do it all remotely, so there'll only be one way to download, and it'll start at 8:00, and IT will record it since everybody's going to be remote, and it'll follow the same format we did with the quizzes and the exam. Now, it is about 11:20 or so. If you feel that you have more questions or would like to go over more questions, whether it be about the quizzes or the or any of the material, we could break out into 3 breakout rooms just to allow you to peruse the information and ask questions, or we could stay as one room. And I will be, while we got still a good number of people here, I will be in the reading room if you have further questions about your quizzes, from 1 to, I'll say 1 to 3, if you wanted to have additional questions. I think Dr. Jarrett answered the questions, most of the questions on quiz 4. So what do you think, Carla? You want us to just break out into rooms so that if they have questions over the next half hour or so, they can
Sure,
peruse any questions they may have? That
sure.
way they'll have the benefit of all 3 of us. If you have any questions at all, feel free. This is why we're here. If there's a slide you don't understand, or anything, it's fair game for anything since the midterm. We'd be more than willing to help clarify or use our knowledge to help you understand these organs.